question, simply choose your answer and hit the Submit button. Today's presentation is Using PK/PD Modeling and Simulation for Pediatric Drug Development, Regulatory Insights into Practical Issues. Our speakers are Nastya Kassir and Justin Pittaway-Hay. Justin is a PK assessor at the United Kingdom's Medicine and Healthcare Products Regulatory Agency. He is a member of the EMA's Modeling and Simulation Working Party. He has over 15 years of experience in clinical drug development. Justin earned his PhD in pharmacology from the University of Adelaide in Australia. Joining Justin is Dr. Nastya Kassir. She's currently a senior director at Certara Strategic Consulting. She joined Certara in 2008, and she is a scientific and regulatory expert for drug development programs for small and large molecules, particularly in the therapeutic areas of pediatrics, orphan diseases, inflammation, and oncology. She's authored or co-authored more than 60 abstracts and manuscripts in the field of PK/PD. Nastya holds a PharmD and a PhD in pharmaceutical sciences from the University of Montreal in Canada, and she's a fellow of the American College of Clinical Pharmacology. Justin and Nastya, welcome to today's event. I'll now turn it over to Justin to begin the presentation.
Thank you, Susanna. So thanks for the introduction. As mentioned, I'm a member of the EMA's Modelling and Simulation Working Party and a PK assessor at the MHRA, the UK's regulatory agency. So modelling and simulation is having a greater role in drug development. It's becoming crucial in some areas like pediatrics, where an extrapolation approach is used. So the Modelling and Simulation Working Party supports the CHMP, the main decision-making committee for medicines approval in the EU, the SORP, so the Scientific Advice Working Party, and the Pediatric Committee as well as other working committees at the EMA. So much of our work is referred to us by these EMA committees, and in doing so, we help them reduce uncertainty in risk-benefit decisions and help them improve product labelling. So as you can see from the pie chart, a significant amount of the work that the MSWP does is related to dosing, with approximately 1/3 of this looking at dose selection, and this is primarily in adults, and approximately half of this is related to pediatrics and looking at dose finding and extrapolation. So we're going to go to our first Menti question for this session. We're going to be asking what your background is, so I'll pass this over to Susanna. If you could just show the menti.com. Yeah. So if you could please enter the code 307168 and just describe what your background is, and this will help us get a feel for who's listening to the webinar. So you can either answer whether you work in academia or you work in industry. Hopefully one of the best of those two choices if you don't. And so we'll just have a look at the answers that are coming in. So it's looking like most people are working in industry. We'll give you just a few more moments just to connect and to answer the question. It's good to see it live though, that it evens up a little bit as well. Once we've got to, hopefully we'll see this, okay, it's starting to slow down a bit. So it looks like approximately, okay, no. Due to the low subject number, of course, these statistics change quite significantly quickly. Once we get to about 30, we might go back to the webinar. So it looks like approximately two-thirds or so are working in industry and the rest in academia. Okay, thanks for that, Susanna. We could go back. Great. So one of the remits of the MSWP is to develop and communicate standards for the design, conduct, analysis and reporting of modelling and simulation according to the level of regulatory impact. And as such, the Modelling and Simulation Working Party released its first 3 Q&As back in November 2018, a year ago. And these were related to presentation of results, allometric exponents, and how to include ontogeny into models. And here at the bottom of the screen, you can find the link to these Q&As on the EMA's website. So today's webinar is going to go through these Q&As and present the EMA's perspective on them, or the Modelling and Simulation Working Party's perspective on them. But what's really great about this is that Nastya from Sitara will also present some examples and how this is done at Sitara as well. Okay, we'll just go quickly into the next Menti questionnaire. Yes, Susanna, if you don't mind bringing that up. And this one's just asking participants whether you're familiar with the EMA's Q&A. And it looks like we already have some answers to this. And again, we'll good to see though that well, we're talking to an audience that may not be completely familiar with the Q&A. Some people are, which is also good to see. Okay. But it looks like most participants are not familiar with these Q&A, but hopefully we'll make you aware of them and give you a good oversight of what is involved in them. Okay, thanks, Susanna. If we go back to the slides. Okay, so we're going to touch on the first Q&A. This is about presentation of results and predictions. So how should the results and predictions of a pharmacokinetic analysis be presented to facilitate decision-making about the adequacy of a proposed dosing regimen in pediatric patients? So when we're looking at these studies and the population PK modelling, the aim of the exercise is, of course, to define a dosing regimen in pediatrics and potentially target a concentration that's been shown to be safe and efficacious in adults. However, this is not always the case, and we'll go into that in a little bit more detail later on. So the population PK model, or the PBPK model, for instance, should be adequate for the intended use, of course, and it should be fit for purpose. We know that the most relevant covariate in pediatric patients is weight, and in particularly younger patients, it's maturation. And one of the things we do ask for when presenting results and it's to be presented for regulatory purposes, is that if patients who are under the age of 1, then exposure versus body weight and age should be depicted in additional separate figures focused on children between these ages. And of course, 0 is set here as a sort of arbitrary limit, but of course, preterm infant, that of course includes preterm infants as well. So here's the first example that's shown in the Q&A. And this is an example from brivaracetam, which was given. So what you can see here is the predicted steady state given by weight on the left-hand side and age on the right-hand side. And you can see it's a continuous scale at the on the x-axis there. And so this is showing at least for this example, it's showing the final population PK model for children using an NHANES database using 2 different dosing regimens. So the top dosing regimen gives as a 2 milligram per kilogram twice a day, and the cutoff is for under 8 years. And then for patients above that, there's a lower dose given. However, for all patients, there's a maximum 100 milligrams twice a day dosing. And the bottom set of graphs, you can see just a flat dosing for all patients, and this also has the maximum 100 milligrams. So the blue line shows the median simulated pediatric steady state concentration, and the blue area includes 90% of the simulated patients. The grey band is the simulated adult patients receiving 100 milligrams twice a day. So, as has been asked, the relevant predefined exposure metric should be presented graphically versus body weight and age on a continuous scale. Here, for instance, it's shown as the steady state concentration, but it could be anything. It could be AUC or it could be Cmax. And this example here clearly shows that for the 2 milligram per kilogram patients who get it for all patients, so the top graphs, sorry, for the bottom graphs, I should say, that leads to higher exposures, especially for the patients in the range of approximately 30 to 50 kilograms, but also for patients who are above the age of 8. Ultimately, a different dosing regimen was actually used in the final label, so in the summary of product characteristics, and there was a minimum dose, minimum age that this drug can be used is actually for 4 years, and there's actually a cutoff limit of 50 kilograms where it changes to a flat dosing of 50 milligrams per day as an initial dosing regimen. But the main take-home message from this graph is just the way it is presented, not particularly the details. So also when presenting dosing, if weight band dosing is being used, then what the recommendation from the MSWP is that the exposure ranges predicted for the different doses of the subsets of the pediatric population should be visualized as box plots. So here you can see different weight bands, so for 10 to 20 kilograms, 20 to 30, 30 to 40, and then 40 up to 60. And then again, similarly to the previous graph, you can see the PK metric for the exposure. So again, that could be Cmax, AUC, whatever. And in the background, again, you see a reference range for the potentially the adult population, but it could also be another population where some efficacy data has been obtained, or you know where the target is. And here you can probably surmise that at between 40 and at 40 kilograms, there's a dose change, and that's why the dose seems to normalize back down to the adult data. And the last request for this when presenting results from an allometric scale, from an exercise like this, is to show the recommended dosing regimen should follow the function described in the PK in pediatrics. So this one compares the doses resulting from the underlying function, and that compares it to what the weight banding is. So in this case, the green would be the most preferred option, where there are smaller dosing steps. Of course, that has to be taken in consideration of other issues such as formulation, of course, as well. But at least you can then see the relationship between the weight band dosing as well as the principle underlying the model. So this is whether it's allometric or mg per kilogram. Okay, so now I'm going to hand this over to Nastya to provide some examples of how this is done at Satara. Thank you, Nastya.
Thank you, Justin. So in order to show examples regarding what Justin talked about, so I'll be presenting results of simulations for a drug. So we're not showing the PK of the drug here, but the pharmacokinetic of a drug was evaluated in adults using population PK analysis. Both clearance and volume of distribution were found to increase with increasing body weight according to an allometric relationship with estimated exponent values higher than 0.7. The objective of this analysis was to support dosing rationale in pediatric patients 2 to less than 18 years of age via pharmacokinetic and efficacy extrapolation from adult patients. The extrapolated exposure endpoints included area under the concentration time curve at steady state, AUC, and maximum concentration at steady state, Cmax. I will be also only showing graphs related to AUC just for sake of time, but it's the same thing for Cmax. In the first step, AUC and Cmax were simulated for the body weight 10 to 100 kilogram in 1 kilogram increments at various dose levels to select the appropriate body weight range for potential pediatric doses. Then, the dosing in pediatrics was determined using an AUC-matched approach. The appropriate dose levels for pediatric by body weight range were identified to match the reference simulated adult AUC within 20% difference. In this case, a 20% difference from adult AUC was selected, but it could be different based on your case and on the therapeutic range of the drug. So here, the median AUC in adult is represented using the red solid line, and the 20% area is represented by the green shaded area. For pediatric patients between 10 and 20 kilograms, sorry, I'll just use my pointer. Between 10 and 20 kilogram, a dose a dose of 1.4 milligram per kilogram was selected, and for patients between 20 and 40 kilogram, a dose of 1.2 milligram per kilogram was selected, which kept the weight-based dose level constant over the body weight range, so that the deviation in median pediatric AUC was within 20% difference of the adult AUC. For the dose of 1 milligram per kilogram, the median simulated AUC in adults and the targeted window, 20% difference from the median, were determined and are shown in dark gray area on the plot. The light gray area on the plot represents the 90% prediction interval of simulated AUC in adults. The AUC range in adults was used as a reference to guide the further verification of the selected pediatric dose. As demonstrated in the figure, median values of simulated AUC at the selected pediatric doses for each body weight range were all within 20% difference relative to those simulated in adults. Another way of presenting the results is, instead of presenting the continuous values of the exposure, is to present the fold change in exposure versus adult median. This could be sometimes easier to interpret, but this is exactly the same data where we see the fold change, and it's all within 20%, so 1.2 to 0.8 of adult data. After comparing median exposure, we are now looking at the distribution of simulated exposure according to the selected dosing regimen in each age group. As you can notice, in each in each age group, there is an overlap of the weight between the different age groups. So for the first age group, which is 2 to 6 years old, the weight range is 10 to 26 kilogram, which means that patients in this group could be getting different doses. So the dose for subjects less than 20 kilo and those higher than 20 kilo will be getting the higher dose, which is 1.2 milligram. The distribution of simulated AUC for the different age groups was comparable to that simulated for adults, so this group, and also was similar to that estimated in adults in the study of interest, and this was also compared to the AUC in adults for the responders, like only responders for the primary efficacy endpoint. And this could be something of interest if you have efficacy data and you want to make sure that exposure in responders is not different than exposure in the rest of the population. In addition to showing the plots of exposure distribution, the percentage of subjects with simulated exposure above 95% percentile of adult exposure were determined with attempts to make it less than 20%. And in this case, they were all less than 10%. This example showed dosing recommendation based on weight groups, but it could have been done based on age group and also presented the same way. After those levels were selected from PK simulation, they were used for efficacy simulation. The model predicted a trend between age and the probability of achieving efficacy endpoint is graphically compared with the observed adult data to evaluate the robustness of the model. The simulated efficacy response in pediatric age groups are also compared with the observed response to placebo in adults to assess the potential treatment benefit. The blue line and the light blue shaded area represent the model predicted mean response and the associated 95% confidence interval in patients at the selected dose levels, So 1.4 for 10 to 20, 1.2 for 2 to 40, and 1 milligram per kilogram for higher than 40. So those are simulated. And on the other side, these are the observed data in adults. We also have the dashed red line and the light red shaded area represent the observed response and the associated 95% confidence interval in placebo-treated subjects. In each pediatric age group, the symbols and error bars represent the model predicted mean response and its 95% CI. In each adult age group, the symbols and error bars represent the observed response and the associated 95% CI. This might not be the best efficacy examples that sponsors like to see, like efficacy decreasing with age, but in this case, this was related to the effect of age-associated risk factors affecting the efficacy response. The age-dependent change in efficacy observed in adult patients was well described by this model, and the simulations suggested a treatment effect statistically in favor of the drug over placebo for all pediatric age groups, with the lower limit of the 95% CI for the predicted mean probability in treated pediatric patients above the upper limit of 95% confidence interval for the observed adult placebo data. Now, after we talked about the work, we can mention that this work for the work related to the population PK analysis can be performed in softwares like Phoenix NLME or NunMem or Monolix or any other software. The simulations for pediatric dosing could be done in Trial Simulator, in R, or any other similar software, and also presentation of data can be done in R or any other tool. And now I'll pass it again to Justin to talk about fixed or estimated fixed exponents.
Just before we go into the next question, we have just one last Mentimeter question. Just Susanna, if you could, great. And again, this is just asking whether you're involved in modeling and simulation for regulatory submissions, just so we can get again a feel for the audience members who are listening in. So it seems at least the majority are involved in regulatory submissions and in modeling and simulation. It's good to see, and hopefully this is also helpful for those people who are not involved. But it's okay. Thanks, Susanna. So quite interesting results there. Okay. So we're going to move on to the next Q&A from the modeling and simulation working party about whether estimated values for allometric scaling exponents should be used in pediatric pharmacokinetic models. So I've just shown here, of course, the seminal work of Kleber, who looked at 13 animal and mammals and concluded that the value of the allometric exponent should be approximately 0.75. However, of course, there's been a lot of discussion in this field about whether that is the correct value or not, and we know that there are other exponents, especially if you're looking at GFR or glomerular filtration rate or blood flow, and other values are used. But the other reviews have shown that it's one of the most commonly used, and it's one of the more widely accepted values in a range of different plausible values. And so we do know that there is a physiological basis for using 0.75 for clearance and 0.1 for volume of distribution. We also know that in practice, and I think Nastya will show this in one of her examples, that sometimes changing these exponents and using the different exponents will not always result in a substantial consequence. However, we do believe that 0.75 for clearance and 1 for volume is useful for the prediction of drug clearances in children, primarily over the age of 5 years. However, what the Modelling and Simulation Working Party have is essentially a preference to use fixed exponents, because we know that these are scientifically justified and practical when developing population PK models in children. However, we're not closing the door on others, but we do say that if they are used, then they should be scientifically justified. And the preference is, though, that if exponents are estimated from adults' data, then these should not be used. So these are, and I'll show you, sorry. And the last couple of points about when using these estimated exponents is that in younger agents, of course, it's important to include relevant maturation functions, and that when developing the models, that the maturation functions and the allometric exponents should not be estimated simultaneously. I'm now going to hand over back to Nastya to provide some examples of how Satara performed these. Nastya.
Thank you, Justin. In this first example I'll be showing, I took exactly the same drug that I showed earlier, where a milligram per kilogram dosing was used. So we will start with the left plot, where we derived the dosing based on estimated exponents of 0.8 for clearance and 0.7 for volume of distribution. Based on the final model with the estimated exponents, the dose recommendations were, as previously said, 1.4, 1.2, and 1 milligram per kilogram for the different weight ranges. When the final model was run with those fixed, we took the final model, we ran it with fixed exponent of 0.75 for clearance and 1 for volume of distribution, and we looked at the recommended doses, and we can clearly see that the dosing recommendation are the same based on the model with fixed or estimated exponents, and in both cases median values for each age group or weight group were within the 20% difference with regard to the median AUC in adults. So in this specific case, using fixed or estimated did not have an impact on dosing recommendation. However, in the second example, I took a drug where the dosing was a fixed dosing, and the initial recommendation were done using the model where exponents for weight were estimated, and these were 0.6 for clearance and 0.9 for volume. And the dosing recommendation using this final model were like 1,500 for 10 to 25 kilograms, 2,000 for 25 to 40, and then 3,600 for patients above 40 kilograms. In this example, we already see that, patients between 40 and probably 46 kilograms were outside the 20% acceptable difference with adults, but that was acceptable. And then we took the final model and ran it with fixed exponents, and based on that, we see that the range or the proportion of subjects outside the 20% increased between 40 and 50 kilograms, and also for subjects, so now we have subjects with a 1,500 milligram, subjects between 10 and 15 kilo who are also outside this 20% acceptable difference. In this case, an evaluation should be made to make sure that the proposed doses does not result in high percentage of patients having exposure higher than those estimated in adults, and to select a dose that could work in both cases. And now I'll hand it back to Justin to talk about ontogeny and maturation.
Thank you, Nastya. So the last Q&A that the MSWP addressed was, how should ontogeny and organ maturation and the impact of changing weight or maturation during a study be implemented into models? So we know quite well that there's a lot of factors that need to be taken into account when developing PK models, and especially PBPK models, or physiologically based PK models. We know that there are increases in body weight and body size and growth. There are changes in GI function, so changes in pH, volume, transit times, and bile secretion, as well as differences in tissue composition. So plasma proteins change throughout childhood, as well as body water and fat. But what we focus on today will be changes in clearance factors, so especially renal, biliary, but especially metabolic clearances. And we know that these are related to body size, but also related to maturation functions as well. So here's the graphic that's shown in the modelling and simulation working party's example. This is just showing glomerular filtration rate, or GFR, measured across approximately 10 years, and you can see it changing over that course of 10 years in pediatrics. When we look at CYP ontogeny, then we know for all the different CYPs there is a different way that these develop as a child, not from birth, or at least even before birth, but from birth generally up until approximately the age of 8. And you can see that this is different for different CYPs, of course, and so it's important to characterize how a drug is metabolized in the models, of course. That said, of course, this graph here shows a mean line. Of course, there is some variance about it, and Nastya is going to show a good example of this, especially related to CYP3A4 later on, where, at least according to the literature, this might change, of course. But we do need to take this into account, and especially for children under the age of 2. So back to the question, so how should we incorporate this into models? So we know that maturation is time dependent, and it can't be explained by using allometric scaling. We know when developing PK models, we know that significant changes in weight should still be accounted for. So this means that if a study is done over in the neonatal stages where there's a lot of catch-up growth, then those significant changes should be accounted for, especially if that's going to have an impact on dosing. We know that maturation functions are generally described well by sigmoidal Emax or Hill equations. However, there are some limited examples of where a linear or exponential factor can be used. And when using maturation functions, it's important to take care when bridging from the source population, generally a healthy population, to the target population. So if the underlying function is coming from a healthy population, we have to make sure that that is relevant for the target population that we're looking at, because there is a potential that disease progression may have an impact on maturation. And the last point, which Nastya touched on before, was for PK/PD models where there's a maturation function should be included, then you need to take care that developmental maturation is also considered. For instance, for her example, where the younger children were less sensitive even for the given exposure, that needs to be taken into account. And we need to make sure that assumptions of equal efficacy with similar exposure should always be considered. And this is especially true as generally sometimes PD endpoints are sometimes easier to collect than PK endpoints in neonates. I'll hand this now back over to Nastya to discuss this from a Sitara's point of view.
Thank you. So as Justin explained, growth and development are the two major aspects in pediatrics not seen in adults. And the PK models based on adult data can be customized to account for organ maturation and for size. So size is the primary covariate for pharmacokinetics and can be referenced using a coefficient of 0.75 for clearance and 1 for volume. The equation of clearance can be customized also to take in account the size effect, so allometric function, the maturation function, and also the organ function. The size effect is usually used with the fixed exponents, but more and more regulatory agencies are asking to see the models driven with both the theoretical, so the fixed, and the empirical estimated exponents in order to compare. In addition to size in patients less than 2 years, maturation needs to be taken in account. The information about organ maturation are available in the literature. In this example, we're showing a plot taken from the paper published by our colleagues at Certera, Samir and Muksasi and J.F. Marier. In this plot, we can see a typical maturation of glomerular filtration rate as a function of postnatal age, expressed as a percentage of adult GFR, where we see that adult values are reached around the age of 2 years. At the age of 1 year, approximately 90%, pediatrics have 90% of the adult values. And here we have an illustration of the separate effects of size using the allometric function. So to the left, the first plot to the left, the maturation of the renal function, the plot in the middle, and the combined effect of both size and maturation, the plot to the right. We can see when we combine the effect that we have a steeper slope for patients less than 6 months, and then a slope more comparable to the size effect alone for older patients. And here we have an example where the allometric function and the maturation function were used together, were combined to provide dosing recommendation for neonates. And based on this work, and after combining those effects, it was recommended that for patients less than 2 years of age, the dose should be reduced and or the frequency also should be reduced. We can clearly see the clearance being lower in younger patients. And in addition to the maturation of the renal function, there are information in the literature regarding the maturation of the liver enzymes in neonates. This plot this plot was taken from a paper published also by colleague at SIMSIP, Farzani Salim, and it shows the relationship between the ratios of unbound hepatic intrinsic clearance in pediatric subjects relative to adult values for CYP3A4 mediated metabolism as a function of age. This plot compares data from different published papers, and the best fit to the CYP3A4 data was shown to be described by a monotonic increase in enzyme activity up to adult values levels by 71 weeks of postmenstrual age, which is almost 7 months, and is described by this equation we have to the right. And the best fit in this plot is the one in a gray, solid line gray. This should be taken in account if simulations are performed for neonate for a drug involving this metabolic pathway. And to summarize, population PK models are developed in adults, and these can be customized to provide information for pediatric studies and for simulations. So after taking in account organ maturation, after taking size in account, organ maturation and so maturation of renal function, maturation of enzyme function should be taken in account for patients less than 2 years of age. And now I'll pass it back to Justin for the take-home messages.
Great. Thank you, Nastya, for those really good examples as well of how to incorporate maturation into the models. So this is just going to wrap up the webinar now with take-home messages. So as we've said, the modeling and simulation is playing an increasingly important role in drug development, and especially pediatric drug development and regulatory submissions, where in pediatric drug submissions, it is nearly always crucial that we use modeling and simulation. So the results of simulations, whether they're being submitted as PIPs or in marketing authorizations, then they should be, the results in the simulation should be clearly shown, and the exposure should be given over the different age and weight ranges provided, and especially for patients under the age of 1 and even 2 years of age, then they should be shown in quite a lot of detail where we know that there's going to be a lot of changes occurring. For models that use allometric exponents, then the preference is to use 0.75 for clearance and 1 for volume, and these should be used. However, this is not to exclude other ones being used. However, there is a preference for this to be demonstrated in the first instance. And as just mentioned now, the maturation functions, in addition to allometric functions, should be taken into account, especially for younger patients, so those under the age of 2. Okay, so we're going to wrap it up and then open it up to questions now. So if you have any questions about the presentation, they'd be certainly welcome to ask them now, please.
Great, thank you so much, Justin and Nastya. As Justin mentioned, we'd love for our audience members to pose questions to our speakers in the Q&A box. You can just type your question in and select all panelists, and we will answer as many as we can. So it looks like we have our first question. It's for Nastya. What is the minimum age covered by the use of the renal maturation function?
Yes, this is a good question. Thank you. The papers the data that were used to develop the equation for maturation function, the renal maturation function, use PMA, so postmenstrual age as low as 26 weeks.
Looks like there's a question for Justin. When should the pediatric investigation plans be submitted to regulatory agencies?
Yeah, no, it's a good question. So the timings of submissions shouldn't be any later than when the end of the healthy subjects or the patient PK data has been collected. So those initial tolerability studies, that's the time when the pediatric investigation plans should be submitted. They definitely shouldn't be submitted after initiation of the pivotal trials or any of the confirmatory trials. So, and especially so during those early phases of drug development.
Got a question for Nastya. We may need a little quick clarification here. Why are the maturation function and allometric scaling not added to the rate constant of absorption?
Yeah, they are actually, so in the cases we presented, they were not added, but they are when, there are cases, of course, where we test actually. I would say we test if they make sense or not, and they are not always added. So if they make sense, they are. Otherwise, we focus on clearance and volume, maturation and allometric function on clearance and volume.
Question for Justin. How much detail on your modeling and simulation should a sponsor present in their PIP? Also, is a formal report required, formal modeling and simulation report required as part of the PIP?
No, a good question. So there are some guidelines on the EMA's website of what should be presented and there are key finding elements that need to be addressed when developing a PIP. That's included there. However, sometimes based on that, if not enough detail is presented, then sometimes the PEDCO, sort of via the modeling simulation working party, will come back to a applicant and ask maybe some further clarifying questions there. Regarding the second part of that question of whether a formal report is required, so the PIP describes the studies that will be performed later on, And of course, the PIP can be also changed throughout the course of development. However, the initial PIP is submitted during the early stages. However, based on further data that might be gathered throughout the drug development process, the PIP can then be amended based on that information that's gathered.
All right. Looks like we've got a couple questions for Nastya. How do we account for the saturation of enzymes in the pediatric population if the drug exhibits not a nonlinear pharmacokinetic profile? And they give the example of phenytoin.
Sorry, I'm just looking at the question. So I don't think the saturation of enzymes, as part of this fixed, or, when we talk about allometric or when we talk about the maturation function, I don't think this is taken account, but this is a good question. I will make sure to look at this in details and get back to the person who asked it.
All right, got another one. When you're doing a population PK modeling approach, should the fixed exponent of 0.75 for clearance be applicable to intercompartmental clearances as well?
Yes, so this is a good question too. Yes, we usually, if we are fixing the exponents for clearance, we should use the same exponent for the intercompartmental clearance too. So 0.75, and if you are estimating them, we should be estimating the same exponent for clearance and for intercompartmental clearance. We usually don't have enough data to estimate different exponents for each of these parameters. So it's the same exponent, fixed or estimated for clearance and intercompartmental clearance, and the same applies for the volumes too.
Someone wants to know, if a drug exhibits nonlinear clearance in adults, can you still apply a fixed allometric exponent of 0.075?
Yes, so for nonlinear clearance, we usually apply the allometric function on the Vmax, the maximum elimination rate of 0.75, or we estimate it too. But usually we only apply it on the elimination rate for nonlinear clearance.
Looks like we have one final question, Nastya. Does the FDA, and Justin, please feel free to chime in as well, does the FDA have the same recommendations for fixing or estimating weight exponents?
So there isn't really a clear recommendation, so it's the same as the EMA, and in many cases we end up estimating or fixing. So depending on the case, sometimes we only estimate the exponents and do all the work, as the example I showed, using estimated exponents, and sometimes we have to fix the exponent and compare if there are, especially if the estimated exponents are very different than what the fixed one, or what is recommended, or what is published in the literature.
Looks like there's one more question.
Perhaps to add there.
Go ahead, Justin.
Oh, sorry, Susanna. Recommendations. I think I'm not aware of what the FDA. I haven't seen any recommendations coming from them, but in discussions with them, that they would accept both as well, taking fixed and estimated as well. I mean, the EMA is also open to looking at estimated or allometric exponents if they are quite a distance from the fixed ones.
Looks like we have one more question. Someone wants to know if you can elaborate on how you incorporate organ function into models? And I'm guessing they mean hepatic and liver function. Or sorry, renal and hepatic function.
Sorry, can you repeat, Susan?
I think what the person's getting at is, should there be, you have a reason to think that the pediatric population that the drug is going to be prescribed for, that the target population might have hepatic or renal impairment, how would you incorporate organ function into your models?
So I don't think there is a difference in implementing the allometric function or the maturation if there is a risk of pediatrics having a different response in hepatic or renal impairment. So if this was not proved in adults, I think, and because usually we develop the model in adults and you have already a proof that there is an effect, a different effect for this population, and then you take this effect to pediatrics in addition to allometric function and to maturation. But if this was not already proven in adults, I'm not sure, how this could be like, estimated for adults, for pediatrics without any information from adult data.
Okay, great. Thank you so much, Justin and Nastya, and thank you to our audience for all their great questions.
Thank you.
Thank you.
The next webinar in the series will take place on November 26th when Atlanta Kassir will present Effectively Developing Cutting-Edge Mobile Tools for Market Access. On December 11th, Roman Cassiano will present The Rare Disease Drug Dilemma, Great Value, High Price, Limited Access. Finally, on December 19th, Drs. Peter Kilford, Mian Zhang, and Yannick Wadagadera will present What's New in the SimCip Simulator. You can register for all of these webinars in the series by visiting certara.com/webinars. On behalf of Certara, I would like to thank you for attending today's presentation. This concludes the webinar. Goodbye and have a great day.